it's my great honor to introduce like the Dr. Michael Lenardo from the NIAID. So actually, many of you, if you know me, you're probably wondering why I'm here. I spent 6 years in Mike's lab. It is my scientific formative year. Learned so much from Mike, as many like the Mike's trainees do. So Mike is like get his bachelor degree from Johns Hopkins University. Then he get his MD from the Washington University, St. Louis. So later he did a brief like the clinical fellow, and then moved to the White House Institute, did the postdoc with Dr. David Boddimer. In 1989, he joined the NIH and started his work. Mike's work is focused on the T cell. So like he focused on the survival and the death of the T cell. He used like the contemporary genetic approach. Actually, he is the pioneer of this to dissect the like the molecular mechanism for this complicated like the immunology disease. So I still remember one in the Mike's lab. The first thing is we get the like the patient's sample to try to figure out what's wrong with this T cell. So I think today Mike will share us with like his recent work on this direction. Please, Mike. Oh, okay. One more thing I forgot. Mike is really great like the guitar player. So he is like I like to share. Could I share something?
Hey Mike, can you let yeah.
So this is NIH like the rock band with Francis Collins and other scientists from NIH. And this one is like in 2017, the library union. I take this picture at the Library of the Congress when they are performing. It's really great fun. Okay, that's it.
Thank you, Lee.
Okay. Mike?
Thanks so much, I appreciate it. Okay, can you see my screen?
You probably need to share.
No.
Oh, okay. I must have clicked out of sharing. Hold on one second. Okay, can you see it now?
Perfect.
Okay, great. Okay, so first of all, I want to just start by thanking Chuan and thanking all the organizers of this amazing international scientific forum. I had no idea this existed until Chuan told me about it, and I think it's really a fantastic way to link together scientists from all over the world around molecular biology and immunology. And I understand that apparently there was some confusion that people might have thought it was yesterday. I apologize if they went there and were disappointed, but hopefully they're on today and we can move forward. So my lab is located at the National Institutes of Health, same as Chuan. In fact, we're in the same building, the big orange building here in the background. And since 1989, our group has been investigating the molecular regulation of the immune system, and with a heavy focus, particularly in the last 20 years, on the human immune system. And in order to do that, we're able to take advantage of this orange building in front, which is the NIH hospital. In that, they have a whole set of clinical programs there that enables us to study children with genetic abnormalities of the immune system. And it really allows us to probe deeply and then know that the actual phenotype of changes in the immune system, particularly genetic changes, what the consequence is for the health of those individuals. And on the right is my colleagues who work with me. As you can see, there's a lot of Chinese, a lot of all Asians. And I just want to give a shout out to Dr. Helen Su here in the purple shirt, who is an independent, very distinguished molecular immunologist at NIH and a fully trained, fully credentialed pediatric immunologist who has won many awards for her work, has discovered a number of really important diseases, and has been a great colleague and friend for a long, long time now, and our labs work together very closely to solve these diseases. So before I get started on my talk, I just want to take a second because I know there's a large number of people here that are of Chinese heritage and maybe perhaps actually in China. Let's see if I can advance this. This doesn't seem to advance. Huh, that's strange. I don't know why the slides are not going. Let's see if we can go here. Okay. And I just want to personally say on behalf of myself and my colleagues at NIAID and NIH, thank you, China, for your scientists' efforts to save lives during the pandemic. You know, there's been a lot of, you know, back and forth verbiage and talk, but I really feel that it's important that we acknowledge the amazingly quick and important steps that were taken in China, because sadly, that's where this virus emerged. You know, we don't know from which animal, but somehow it emerged and struck Chinese people first in Wuhan. And almost immediately, the physicians and scientists there recognized that this was a very serious problem and brought the word out, brought the word forward very, very bravely. And then amazingly enough, within weeks, January 9th, the scientists there figured out that this was a novel coronavirus with samples from those patients and put this out on the web. And I want to emphasize that this is not made up information. Each thing I'm pointing out here, there's the web link. You can check online. Nature Journal reported it was a novel coronavirus, and then very, very rapidly, within days, released the entire genetic sequence online, expressly so that other countries, other scientists could do PCR tests, begin vaccines, and do further investigation because of the importance and severity. By then, already there were tens of thousands of cases. It was spreading like wildfire, and people knew it was going to be extremely dangerous. And then following that up, the joint Chinese WHO commission, and this was now China inviting in scientists from many European countries, from the United States. I think it was co-chaired by a Chinese scientist and an American scientist, to issue the first report on the nature of the virus and the illness. And the one thing as an immunologist I want to point out that was critical from that report was the epidemiology that showed this was spreading in family groups by droplets, so that this was not simply going from animal to human, but now could go from human to human. And that's really the basis of a pandemic. When it can spread effectively from human to human, that makes it very, very dangerous. So I just want to point out that that those actions, you can see the timeline there. It's just spectacular, using all the most current tools of science to put it out there, what was going on, so that other countries could take proper steps. I think that no doubt that saved millions of lives around the world. And also the actions of the government to shut down, to begin to quarantine, so that, you know, there's a lot of travel in and out of Wuhan, to shut that down so that it could limit, as soon as they knew what was going on, to limit it from further spread outside the country and within the country. So I just want to thank you. And you can see the wonderful words from Tu Yu. Her work from saving millions of lives from malaria, I think, was repeated by the brave scientists and who took the steps to bring this to everyone's attention. So thank you very much. Okay, so I want to talk a little bit about the kind of work we do, and this is molecular Mendelian genetics. And what I mean by that is the work that we do is based on the very fundamental principles that Mendel laid down in the 18th century studying peas, but we have enormously powerful tools now to use those concepts to make progress in human disease. And there are all the things you know about, but even as recently as 20 years ago, didn't exist. Next-gen sequencing, high-efficiency transfection methods, RNAi, shRNA, CRISPR technology for gene and genome editing, induced pluripotent stem cells, and most importantly, the incredibly advanced computers like the one I'm speaking to you on here in a hotel room. It gives us incredible power to do genomic analysis. And so we call this Mendel on steroids, and you can see his picture here on the right side of the screen. Any of you who live in California or are from California, you might recognize the youthful body of one of your former governors who was a bodybuilder, and with Mendel's head. So you might ask, why Mendelian genetics? You know, we have these very widely used genome-wide studies that enable you to analyze millions of variants and can try to connect those with disease. And the diagram on the left, which comes from a beautiful article written about 15 years ago by Jim Lupsky, a brilliant geneticist at Baylor, really explains why we did that. And so initially we did work on GWAS identified variants, but they're very difficult to do because they have very small effects on disease for the most part, except for the MHC locus in immunological diseases. And the reason is that it's explained when you think about where mutations are born. So mutations are born by single events that occur in the genome, often during meiosis. And then those are built into a germ cell that then becomes incorporated into an individual. And depending on what level of severity of impact those have on that individual's health will determine how much reproductive fitness they have to pass that allele along. For the most severe ones that either cause death in embryogenesis or very severely incapacitated individuals once they're born, they never will be able to reproduce and have children. And so therefore they will not pass that allele along. So it doesn't even become Mendelian. It just stops as a de novo mutation. But if it is passed along, then the degree to which it impairs an individual's ability to get married and pass along their genes by reproduction will determine how far it gets out in the population. And by definition, the GWAS studies were set up to interrogate polymorphisms that were in the population at the level of about 1%. So what you can see is these then have undergone a process that we call purification by selection, and by definition should not have major effects on the phenotype of the individual. Now, there's all kinds of exceptions. If it's an idiosyncratic drug reaction, it might not show up in terms of effects on reproduction, but will have a big time phenotype when you give that particular drug. So there's exceptions to this rule, but in general, we wanted to go into the Mendelian realm here down in orange and in yellow, because that's where a lot of the big effects are going to be. And indeed, that's proven to be true in the diseases that we've studied. Now, when you do human genomics, those of you who have done this, your lab moves in a very different territory than when you do basic biochemistry or work exclusively with experimental organisms, such as mice or flies or worms. And that is, you have the whole sociology of human beings that is overlaid on your research. And that involves also the human beings, the physicians that care for them, because they now become your partners in this research project. And it brings in a lot of different kinds of personnel. So over the years, my lab has added a clinical group that includes nurses, genetic counselors, physicians, who actually work with the patients and the patient families. And so when on the left hand here, I've just diagrammed what I call the virtuous circle of discovery. I think it's fairly self-explanatory, but I'll just mention a couple of things about it. One is that before you jump in and do sequencing, it's important to do a thorough clinical workup and understand the molecular and cellular phenotype of the immune system, because that really allows you to know what you're looking for, what the genes are how they're going to impact the immune system. We then do the whole exome, whole genome sequencing and computer analysis. And I think the point I just want to make is that probably the most important thing that we've learned about the human genome from whole exome or whole genome sequencing is that we have many, many more variants than classic genetics would ever have imagined that we have. And that complicates genomic research, because what that means is you don't get a single gene variant that's abnormal in most cases when you carry out this sort of analysis of a patient, but you get whole lists of them that you have to prioritize. And that can be quite a complicated process, and it can't yet be solved by computer analysis. And I want to also point out that when you think about whole genome sequence, the annotation of the non-protein coding regions of the genome, we're still really much in the infancy of trying to understand what all that sequence means. There's large amounts of it that are highly conserved. There's large amounts of it that are transcribed, even though it's not protein coding transcription. So I think there's going to be a huge effort over the next decades ahead that will help understand how that impacts the other parts of the genome in human disease. Then there's a process of gene validation and pathway study, and that's where you really do the work, kind of old-fashioned slow science, to sort out which of the variants is really the most likely contributing to the disease. And again, there, we've done a lot. The human immunology framework, research framework, is very rich in pathways and discoveries that have already been made, so you have a lot to build upon. However, looking at the interactions of 2 or 3 genes, how hits in different pathways work together to cause a patient phenotype, again, I think is still very much in its infancy. And also, one of the themes that I'll talk about in the first disease I'll describe to you is how the environment plays out on the same genetic background, causing very, very different diseases and phenotypes in different patients. So we have just enormous things ahead to learn about those facets of the human genome and how it affects the individual's life. And then finally, this is the part where we go back to the family and talk to them about what the discoveries, the implications of the discoveries for their future genomic health and also their future family planning. And then hopefully, if we're lucky, the understanding of the gene in the pathway will enable us to introduce a new therapy that would be not a therapy that could have been guessed just by the patient's phenotype. And the second disease I'll talk to you about will give you a great example of that, one that we're super excited about and has quite a dramatic effect. So the first disease I'm going to talk to you about, it occurred in 2 kindreds, one that was actually identified in a sequencing cohort at NIH, and the second was a collaborative effort between Ahmet Ozan in Turkey, Marmara, Turkey, and Bernice Lowe at the Sidra Medical Center in Qatar. And these were loss of function mutations in a gene called RHBDF2, which encodes a protein called IROM2. These individuals had strikingly different clinical phenotypes. So in the case of patient 1, this was an offspring of consanguineous parents with a brother and a father that were homozygous for the mutation. This individual, the younger brother, was homozygous, and this caused a series of extremely severe pulmonary infections that led to them being in the pediatric ICU, which is these dots here shown with PICU. And this led to really very severe destructive effects on the lung. And you can see down here in panel C, P1, that's a computer tomogram of the lungs. And those areas, the big white blob in the middle is the heart on either side. Those are lung areas that in normal person, like most of you, you would just see black, and they would be open to the air with maybe a very fine reticular pattern. And instead, you see big gaping holes, a lot of infiltrates that suggest there may actually be active pneumonia going on now. Now, in contrast to that, patients 2, 3, and 4 in pedigree 2 had no lung disease. They had maybe mild infections during the course of their life, but no severe lung infections. Rather, patient 2 and patient 3 had almost no severe disease that required hospitalization, maybe mild infections. But patient 4, in addition to that, developed a disease that looked like ulcerative colitis. And we have here, these are in panel C, the right two images are images from intestinal endoscopy. And you can see there's ulcers here, evidence of hemorrhage, and abnormal mucosa is what you would see with inflammation of the gut. And if you biopsy those, that's exactly what you find. So this gene is a very interesting gene. It is a member of the rhomboid family of proteins. Rhomboids are normally proteases that occur in the cell membrane to cleave things in the cell membrane. And the IROMs are inactive rhomboid proteins. There's 2 of them, IROM1 and IROM2. And those are also, you can see multipass transmembrane proteins, but these act as chaperones to bring to the surface other proteins. In particular, the most important one for what I'll tell you today is ADAM17, which itself is the protease. And I'll describe the pathway in just a second in the next slide. And this protein is restricted largely to cells of the immune system, which is why we found an immune or infectious phenotype. And you can see the different cell types that show up. And you can see in panel F that this protein is effectively lost by these mutations, which are premature stop codons shown here by the stars in the upper diagram. So this would be a classic recessive loss of function Mendelian disorder. So as I mentioned, these are inactive homologs of the rhomboid proteases that act pretty much as chaperones. And you can see here in the middle that what IROM1 and 2 do, and I should mention here that the association with the proteins as chaperones starts here at the bottom in the endoplasmic reticulum. You can see it grabs the immature version, the zymogen version of ADAM17. It then escorts that to the Golgi, where yet another protease named furin will cleave ADAM17 to cause it to become mature. And then that is then ferried to the cell surface. And initially it thought that that's all it had to do, but actually we now know from beautiful studies from Matt Freeman at Oxford and others that actually IROMs have to stay there. They have to stick around and actually control the stability of ADAM17, or it will be lost from the cell surface. Now, ADAM17 is a protein that came to light in the early 90s because it was recognized that TNF and EGF receptor ligands were synthesized as precursor forms that were membrane tethered. And what it was recognized is that they had to be proteolytically released in order to get the effect of the soluble cytokine. And there are many cytokines, dozens of cytokines that require this, and that ADAM17 and a couple of other proteases were really important for doing that at the cell membrane. And as you can see, ADAM17 here in green is itself also membrane tethered. So this whole process builds a complex at the cell surface, carries out these processing reactions, and then releases the soluble cytokine. Now, in contrast to IROM2, which is restricted to hemopoietic lineages, as I showed you in the last slide, IROM1, and I'm sorry, there's a misprint here. This should be IROM1, is ubiquitously expressed, generally not so much in hemopoietic cells, but in other cells in the body that require this kind of cytokine and growth factor processing to occur. So what is the consequence of this deficiency? And so I just want to show you in the different patients what happens, and I'll focus mainly on the left-hand side of the slide here. So first of all, if you look at the processing reaction of ADAM17 itself, which is shown in panel A, you can see it starts as a mature form and then is cleaved down to a lower, faster migrating, lower molecular weight form. And that form is effectively completely abolished in the patients that lack IROM2. So you can see that chaperone function is absolutely crucial for ADAM17 maturation. And the consequence of that for the patients is shown here in panel B. So these are the 4 patients, 1, 2, 3, 4, right across the bottom. And what this is staining for is the level of membrane TNF. And you can see that in a healthy control, there is some membrane TNF, but it's normally, if you look across a whole group of normals, and I won't bore you with that, it's normally in the range of 20 to 30% on the cell surface. But you can see that jumps up to about 2 to 3 times that level in the patients. And that's not so much true of the sibling or the father, the heterozygous. They seem to still hover down around the normal range, maybe slightly elevated. So it looks like this is really something that is critical to have at least some enzyme function. And if you have some, you might be able to get down around the normal level. And so the consequence of that is shown here. You can see multiple determinations of the patients compared to the controls. It really is quite a different dramatic molecular phenotype. And in addition, what that means is that the release of soluble TNF is substantially reduced, and that's shown here in panel D. So that's in tissue culture. You can also see that same effect if you look at serum TNF that's shown in panel K, that the level of TNF in the serum of these individuals is reduced. And TNF is involved in many different types of immunoprotective innate immune responses and adaptive immune responses. So this is a very, very important consequence. And I won't go through all the data here just for lack of time, but if you do a knockout of the IROM2 gene or the IROM1, and I'll show you a little bit of that later, you can recapitulate this same phenotype. And if you put the gene back into patient cells, you can overcome that phenotype of membrane-bound TNF. So if we want to try to understand the disease, we wanted to try to model what was going on in the patients. And when we compared this radically different type of phenotype, it occurred to us that this was probably not just a genetic background effect, which we've seen cause pleiotropy and other genetic diseases that we've studied, but probably a perturbation by some environmental factor. And so one of the first things we looked at, and unfortunately I won't be able to go through all this data with you, was differences in the microbiome in the patients compared to control. And that showed pretty striking differences. And so that led us to consider the effect that maybe the response to different external microbiome or microbiota could determine the phenotype. And this just shows you an experiment in which we took a knockout mouse with this gene knocked out and then used a pathogen that had been isolated from the patient, Pseudomonas aeruginosa, which the patient on multiple occasions ended up in the ICU, patient 1 that is, from having this. And what you can see is that compared to control mice, the response in terms of loss of body weight, the survival was really compromised by the loss of IROM2. And if you looked at the amount of pathogen in the lungs of those mice, and if you look at the lungs themselves, here, as you can see below is the knockout mouse here. This is just packed with inflammatory cells. This is the wild type who's also sick, but you can see much milder. The impression that you get is that these have really, the mice are really compromised in their ability to handle this pathogen and clear it. And as you might expect from the data that I showed you from the patients, the production of soluble TNF is dramatically reduced. And if you look at the biochemistry of this, you can see that the lung epithelial cells do not have good maturation of the ADAM17 protein, which would be critical for the production of soluble TNF and other factors. Now, one of the things that was so striking about this individual's anatomy were those giant holes in the lung, and those are called pneumatoceles. And one of the critical repair proteins that's made in this same fashion by the creation of a membrane-bound precursor that is then released by atom 17 is a wound healing protein called amphiregulin. And we wanted to know, would the compromise of IROM2 prevent the normal production of amphiregulin? And so what we did was we took a number of cell lines that are lung cell lines, did a knockout both for IROM1 and IROM2, because remember, IROM1 is more prominent in non-hemopoietic cells. And then if you look, you can see here in panel H that the production of soluble amphiregulin is dramatically reduced, as you might predict, by the knockout of IROM1, even more striking by IROM2. So it looks like lung cells, which if you look, you can actually pick them up in single cell RNA-seq, particularly the type 2 pneumocytes, that the release of amphiregulin is compromised. So this led us to try a really clever assay, and this was set up by Jill Fritz and Haley Racky, and has now been used many times by Satoshi Kupo in the lab, that if you take plates of these cell lines and you do a scratch through them, if you wait, that scratch will now, by cell migration and adhesion to the plate, now seal up that quote unquote wound. And so in the control cells that are wild type, that happens within 1 day and is very, very reproducible. If, however, you incapacitate IROM1 or particularly IROM2, you now find that scratch wound does not close as efficiently. But if you add back growth factors, you can actually make it somewhat better. So this is really dependent on those kinds of growth factors that are produced, such as amphiregulin, and this just shows the statistical analysis of that. Okay, I'm not sure what's going on here. It looks like somebody has the ability to put lines on the screen here, so I don't know if they could remove those, but
I think this is some defect with the Zoom software. Sometimes it spontaneously have this scratch. Sorry, I mean, yeah.
That's okay. I think we could probably see through it, but I thought if actually making those, we could we could remove them. But okay, so that gives us a way to think about the first patient, and we need to be looking for patients that might have lung disease due to this genetic abnormality. But what about the colon disease? And so there we again wanted to use the knockout mouse model to try to see whether or not we could recapitulate what is going on there. And to do that, we used C. rodentium as a model for colitis in the mouse strain. And what you can see is, once again, the response of the mouse to that pathogen, the disease is much, much more severe. And you can see here that this is the level of bacteria that's produced. The colon seems to be much more severely affected, with a much greater loss of goblet cells. And again, the very same pattern that you see if you look, actually taking T cells now from the lamina propria of those guts, you can see in the wild type, you get very little membrane-bound TNF, probably meaning that they're releasing a lot of it. And yet you see that there's a very much increased level of membrane-bound TNF in CD4s and CD8s with a knockout. So and then if you look for membrane-bound TNF, you see it, and you also see a reduction in serum TNF. And there's a number of other cytokines that are also affected. So this shows you that really many different disease processes are going to be affected by this genetic abnormality. So in contrast to the classic way that we think about Mendelian diseases, where you get a very specific phenotype collection of syndromes, in fact, when you begin to impair the immune system, maybe a better way to think about it is that you're going to run into genetic defects that, depending on the individual and the specific pathogens that they're exposed to, you're going to get a lot of different pathogen-specific disease manifestations. And I think, so it's important for us now to be on the lookout in our genetically impaired individuals for a lot of different kinds of phenotypes that could emerge depending on the environment that these individuals are exposed to. And I'll just point at the bottom, I won't really spend any time on it, that if you do the scratch test with colon cells, you don't see any problems. So it looks like that's really something to do with the lung in response to injury. So I want to just summarize that we now have a new genetic disease. It's called IROM deficiency with respiratory and intestinal inflammation and cytokine secretion defect, or IRIS disease. And the take-home message is that the mutations in IROM2 or the RHBDF2 gene cause pleiotropic infectious and inflammatory diseases. And this is due to a failure of proteolytic maturation of ADAM17 and defective production of downstream cytokines. And I just showed you data for TNF and amphoregulin. There are a few others on the slides, but actually a bunch of others that are abnormally affected. And if you challenge with different pathogens, you get different phenotypes, but the disease process appears to have the same elements, which is a failure of release of cytokines from the cell surface and also a defective immune clearance of that pathogen. So we will see, as more of these genetic defects with this gene emerge in the future, what the range of phenotypes will be. So now I want to tell you about another disease that we've worked extremely hard on the last few years, originally discovered by Drew Comrie and Ahmet Oz in the lab. And this disease originally was sent to us from an immunology clinic because the children with this disease have recurrent infections. And the reason for that, we discovered right away, was that they have very low serum immunoglobulins, as plotted here in red, and the dotted line up here, shown here in the upper left-hand panel, is the level in normal people. But there were a number of other problems with these kids that were really not very immunological. So they had a history of very severe abdominal pain, nausea, vomiting, diarrhea. And in fact, they were seeming to have such bad diarrhea and such poor nutrition that all the other proteins in their blood, so for example, plotted in black here, is just the serum albumin. And when your serum albumin goes low, the oncotic pressure in the vessels is reduced, and you get all sorts of secondary complications, such as third spacing of fluid and the presence of edema, and really makes these children quite ill. So overall, they were suffering, and this seemed to be right from birth. This was what was happening. So suggesting this was a genetic disease. And if you look at the consequence of this poor ability to eat and feel good about eating, and the loss of protein in the diarrhea, you can see the red lines here are their growth and height and weight curves, which are below the 10th percentile. So these kids are effectively starving to death. And when we analyzed the gut in these kids, we could figure out immediately why they had diarrhea, and that was they had a condition called protein-losing enteropathy due to lymphangiectasia. So lymphangiectasia is just a fancy word for saying inflammation and distension or swelling of the lymphatics in the gut. And what we noticed, and I'll show you a more graphic picture of this a little bit later, that they basically, the lymphatics were incapable of returning the protein-rich fluid from the vessels, from the third space back into the venous circulation. And to top all of this off, what we also found is that in a number of the kids, there appear to be very severe susceptibility to developing blood clots. So this shows a blood clot in the right atrium of the heart that can throw off pulmonary embolus, and that's actually killed a number of children with this disease. And this is a blood clot in the inferior vena cava, which is the big vein that brings blood back from the lower extremities. So this is a really terrible, terrible disease. There are a couple of people that we have found now in a large group of these that we've studied that can make it to adulthood and beyond, into middle and even getting into old age. But most of the children that get this disease die in their 20s. And so it can be an extremely severe, debilitating and life-threatening disease. So what we found is that whole exome sequencing revealed mutations in a protein, in a gene called CD55, which encodes a CD55 cell surface protein that regulates a part of innate immunity called complement. In the old days, this was called decay accelerating factor. And as you can see from these families, this is, again, also recessive loss of function trait that is due to deleterious mutations that are carried in consanguineous families. Many of these are in Turkey, but we have other ones in Syria and Morocco and Thailand. And what these do is effectively eliminate the protein, as shown here by cell surface staining or by a Western blot. So they all share the same molecular pathogenesis and genetic cause. So complement is a really interesting system that we've learned more and more about over the decades. Initially, for example, when I was in medical school, it was thought that its sole function was to use the C1q receptor to recognize antibody-coated microorganisms. And then this would trigger a cascade of proteolytic reactions through C3 and C5, which would eventually cleave a variety of proteins to form something called the MAC, or membrane attack complex, which is a complex of C5b through 9. And that would sit on the microorganism and cause it essentially to pop. However, we now know that many things that trigger innate immunity, damage-associated molecular patterns, apoptotic cell-associated molecular patterns, pathogen-associated molecular patterns, can be recognized by other receptors like mannose binding lectin and ficolin, and these can also drive the complement pathway. In addition to forming the MAC, it also can trigger the production of split products of these factors. C3a and C5 are very notable. They're called anaphylotoxins, and those can bind receptors on immune cells and drive all sorts of cytokine production changes and other kinds of responses that drive inflammation. And what CD55 does is it just directly binds to the C3 convertase and essentially controls the level of activation of this pathway during innate immune responses. So if this were the problem, then you would expect to find in our patients hyperactive complement. And this slide just shows you that indeed this is the case. If you look for various kinds of split products like the C3d, which is particularly stable and sits down on the surface of cells, you can see either in normal media or acidic media, which is called the Ham test, you can see the overaccumulation of complement products in the patients compared to controls. And if you do a Jurkat knockout, you can actually separate CD55 positive and CD55 negative cells in the same culture, and you can see the hyper deposition of the split products from complement in the CD55 knockout, but not the CD55 intact cells. So how does this relate to the gut disease? Well, the way that it relates to the gut disease is if you look on the lymphatics in the patient samples, you can actually see C3 split products, the C5 through 9 membrane attack complex on the lymphatics, and is obviously damaging those lymphatics and preventing the lymphatics from working normally. And so consequence of this is, you can see here, and that is that there's a massive lymphocytic infiltrate in the in the lumen of the gut, I'm sorry, lining of the gut. And then also you can see this is a giant distended lymphatic. And if you look carefully in the edges of this, you can see it's breaking down and just releasing that fluid into the gut. And that essentially is why these individuals have such severe abdominal pain, nausea, vomiting, poor nutrition that's causing them effectively to starve to death. So the dysregulation of complement can directly damage the gut in a severe enough way to cause this syndrome. And it's interesting, and maybe we can come back to this at the end, that this occurs selectively in the gut. Now, one thing I want to share with you, which is quite interesting about human disease, is that we had two families that we presume were unrelated, but they were from the same region of Turkey, but they had the exact molecular genetic mutation. And when we looked into this further, you can see with this massive pedigree that actually they're in the same family if you go back far enough. And this goes back through generations. And when we went out to this region, talked to people there, we found out that the village elders in this very rural area of Turkey said that this was a terrible scourge that's been in this part of Turkey for many, many generations. It was so bad that it has a special name in Kurdish, which is Turdigan, which means cureless or tragic. And they actually had folk remedies out there where they would take the skin of a black sheep and wrap the children in this in an effort to prevent them from dying. And of course, that remedy doesn't work. So that was a really particular problem of why this was so common there. So I went over from NIH with a very small flow cytometry instrument here that I put in my suitcase. That was really interesting to get that, and a bunch of blood tubes through the TSA in Dulles Airport. And we flew over and we drove out to this rural area with a team of scientists from Ahmet Ozen's group in Istanbul. And we set up shop in a little ambulance service there. And we went to the schools and invited the parents to come in and give us informed consent for their children here. You can see. And then the kids who are smiling here, but some of them were not sadly smiling after they got their blood drawn, but we did give them little treats. And what we learned from that is that there's a big founder effect in that area with a carrier frequency of 15% in a lot of affected children. So this really brought home to us the need to do something for these kids and try to stop this terrible disease. So how do you do that? Well, it turns out there was one drug on the market called eculizumab that blocks the complement pathway at C5. And this had been developed for paroxysmal nocturnal hemoglobinuria. But if you look here, when you put that antibody in vitro with the cells, it completely shuts down complement on the patient's cells. Interestingly enough, you can see that normal people also have activation of complement normally. So I think the way to think about this is that complement is sort of idling along to a certain degree in all of us, probably waiting for a pathogen to squelch. But then this just gets to be a little bit more prominent in these kids because they're missing this inhibitor. So I want to show you some of the data here. So this is just the serum values of total protein, albumin, immunoglobulins, and vitamin B12. And you can see for years prior to time zero, which is where we're going to give eculizumab to a variety of these patients and for each of these parameters. And you can see they're way below the normal value, which is indicated by the gray region in these graphs. So if you begin to look, almost immediately after the administration of this, literally within days, all of these parameters return to normal. And it's even more dramatic than that. When you give the shot, literally within hours, the patients begin to say that their pain has gone away, they're hungry, they want something to eat, they stop vomiting, the diarrhea goes away. So it is just quite dramatic, which surprised us because we were worried about damage to the gut that would be irreversible due to the longstanding inflammation. This just tabulates a whole variety of different parameters that we published this year on Nature Immunology. So first of all, you can look at the gut directly with an endoscope. You can see how all these big aggregations of lymph, those are the big lymphatic vessels that are lymphangiectatic. The whole colon looks milky because of the release of this milky fluid into the gut, which is very abnormal. And after treatment, this returns to a normal appearance of beautifully nice erythematous colon in these children. All these different types of really severe abdominal upset and problems, which have been longstanding in these individuals, go away. They still have some vitamin defects that have to be made up over a longer period of time. And then this just shows a whole variety of different medical therapies that were tried with these kids were and ineffective. So basically doctors were throwing every kind of immunosuppressant or different kinds of nutritional supplement at them. You can see that when they get this drug, effectively every other medication is removed from their medical care, and they only need injections of eculizumab. So it's a really, really striking example of how when you figure out the molecular pathogenesis of a disease and have a direct intervention, now their treatment is focused specifically on that molecular problem. So I want to just show you this boy. This is patient number 5, a very sweet young boy from Turkey, who in this photograph in 2017 effectively was dying in this hospitalization. If I didn't show it because it's just so awful. If you pulled back that blanket, you would see that this boy is essentially skin and bones, looks really like a starving victim that you might see in the pictures from Africa or concentration camps. And you can see his hair is really very thin and sparse because of nutritional deficiency. His skin is mottled and due to this. The dentition, his teeth are really terrible. So this boy is effectively on the brink of starving to death when he got back then in 2017, one of the first patients we gave administered eculizumab in the hospital at Marmara University by Dr. Ozen. And here is one of the more recent clinic visits. So you can see that once you stop that disease process, the recovery is global in all the different systems, and he's actually able to recover. So it's just startling, spectacular what happens when you treat the disease. So I want to just stop there since we're out of time and say that this disease, which we call CD55 deficiency with hyperactivation of complement, angiopathic thrombosis, protein-losing neuropathy, Chapel disease, is caused by abnormal complement activation, as I showed you, due to autosomal recessive Mendelian loss of function mutations in the CD55 gene. It's due to reduced CD55 on the cell surface, and it has to be there to arrest the hyperactivation of complement just in the normal exposure to microbiota in the gut to prevent those terrible complications of intestinal lymphangiectasia. The treatment, which I showed you, is that you can use the already FDA-approved eculizumab, and that's a very effective treatment. This is actually patient number 1 here, this cute little girl who is also very effectively treated with eculizumab. We now have several dozen, both in our cohort and other doctors around the world. And because that requires an infusion at a medical office and is restricted in access around the world, we are now working with another company, Regeneron, in a trial of a novel C5 inhibitor. We've actually brought a patient to the NIH Clinical Center from Bolivia with this condition, and he is really done spectacularly well on that medication, and we're hoping that that will be a successful clinical trial, and that there may be other complement inhibitory drugs that could be used for this condition. So I want to stop there and thank a huge number of individuals who participate in this research. The iris disease work was done by a number of people, but really led in the recent years by Satoshi Kubo, an extremely talented postdoc from Japan in our group. And the past year has been working with a very talented post-baccalaureate fellow named Ria Kataria from New Orleans. And I also want to just give a shout out to Jill Fritz and Haley Racky, who now are out of the lab, but they did beautiful work on the early identification of the first genetic variants. The second family, as I mentioned earlier, was from Bernice Lowe's group at Sidra Medical Center in Qatar and Ahmet Oz at Marmara Hospital in Istanbul. And our clinical team really contributes just enormously to be able to do this. I'm not a clinician, I'm a basic scientist. So Helen Matthews, our nurse, Morgan Similak is our genetic counselor, Hugh Jung is our bioinformatics, and I mentioned Helen is an independent PI, but has been gracious to help us do this kind of work in the most effective way possible. And Chapel disease is Drew Comrie and Amit also on that disease. And Khan, Bostag, and Rico already actually helped us by providing the second patient, which led to identification of a number of other patients. And they're at the Center for Rare Diseases, and also a very, very important group in Europe in this kind of research in Vienna, Austria. And of course, a number of other people are also play an important role in this work. So I guess I'll stop there, Chuan, and be happy to take any questions. So sure, go ahead. I see, Chuan, you have your hand raised.
Okay, I guess I'm gonna to kick off. So it's really marvelous study, and it's really striking to see the patient actually get recovered. I mean, it's really spectacular. So I actually have questions, mostly actually related to this, the first part of the talk. I'm actually curious about the co-expression about the ADAM17 with the two proteins you mentioned, which is the RAM1 and RAM2. So how about what's the situation about the expression pattern of the ADAM17 with either one or two? What's the expression, the distribution of the co-expression pattern?
Yeah, so basically, I think the co-expression, the expression pattern of ADAM17 pretty much overlaps with IROM1 and IROM2. I don't know that it's identical, which might suggest that there's other roles for ADAM17 in its unprocessed state, or there may be other pathways of processing. But I think that in general, when you think about the maturation of ADAM17, you're usually talking about pathways in the same cell where there's co-expression of either IROM2 or IROM1. Now, IROM1 is in many, many cells in the body. IROM2 is less restricted, and in general will be in cells where there is no IROM1, and those are mainly the immune type cells. But that in broad brushstrokes, that's the general patterns that we'd be talking about.
I see. So I think my second question is, I noticed that one of the striking phenotype from the first part is the cleavage of TNF. But is that actually particular? I mean, I'm not sure whether this dominantly from the epithelial cells, the TNF cleavage, or it's actually also related to other immune cells, for example, like T cells, the deficiency.
The deficiency of, you're talking about IROM2? Yeah. The deficiency of IROM2 is complete throughout the body. And the reason for that is that the mutations are premature stops. And the effect of those is to cause nonsense mediated decay of the message. So the message is dramatically reduced by that system. And so when you look for protein, it's effectively gone. I think if in any cell that we've checked, it should be gone.
So it's universal.
Yeah, we think so.
I see. Okay. My third question is, so because the patient symptom or phenotype, we observed that at least the 4 patients you showed, that the number 1 and number 4 patient actually have the most dramatic phenotype, while, I mean, in terms of the lung and then the intestinal phenotype, while the number 2 and number 3 is not that obvious. So my question is, because the either the rescue experiment or the cytokine expression experiment showing that all these deficiency related to the cytokine expression problems. But I'm wondering why you think this difference between the patient's symptom, I mean, particular patient number 1 and number 4 compared with number 2 and number 3.
Yeah, that's a really, really good question. And this is a thing that we often run into in diseases, but more commonly, it's in diseases you see in say, like the developed world, like the United States. So in the US, the type of genetic mutation you normally would see in the immune system, or that you see more commonly in populations that are outbred, that don't have consanguinity, are frequently haploinsufficiency or dominant interfering mutations. So examples of those would be, for haploinsufficiency, CHI disease, which is CTLA4 haploinsufficiency. So they can get sick due to just loss of one allele. And also another disease like this would be ALPS, in which a mutation on one allele can cause a dominant interfering molecule that will screw up the overall FAS, a death-inducing complex. So in those cases, to see unaffected family members is actually very common. So in CHI, I think what we call the penetrance of disease, meaning you got the mutation, you got the disease, is only about 50%. In the case of ALPS, our 20-year study showed that it's about 70%. So that means you've got a lot of people out there that are relatives of someone that has disease that is unaffected. Now, there are probably a number of reasons for that. One reason we've explored are other genes in the background. So it could be a 2 or 3 gene hit that now gives you the full-blown disease, whereas if you only get the FAS hit, it's not enough. But I think there's also going to be environmental factors. In this instance, in iris disease, it was quite surprising to us to see that phenomenon, because this is a loss of function mutation, and it is homozygous. We normally don't see that. We normally see a much higher level of penetrance, but you would say here our penetrance is low. It's basically, essentially 2 non-penetrant out of all the patients we have. So it really says that there's some other important factor that has to drive disease, and that's why we started thinking about the microbiome, which is probably the biggest part of our environment that affects us, right? Because it's with us all the time. It can change. It's on every mucosal surface of our body. So it's a really important question. I don't know that we know for sure why those individuals aren't infected. They're very lucky that they're not, because it's not a fun disease to have, either version. But we could imagine as they go through life that they're going to be susceptible, and they could get hit anytime with a bad pathogen that they can't clear because they can't make soluble TNF, and then they're going to be in trouble. And also, I think it points out another aspect of the sociology of human investigation, and that is, we haven't been with these individuals for their entire life. The doctors they have now may not have been there when they were 6 years old and maybe got a really bad infection, and they just don't remember it. It didn't make an impression. They got better. They attributed it to something else. So you're limited by the information you have, and we can envision in the future, and maybe China will be a leader in this, or other countries that have a more nationalized health service, where the electronic medical records will give you a really accurate 100% picture of what the health of that individual has been, at least in terms of their interface with the healthcare system. But right now, around the world, even in the United States, healthcare records are a mess. I mean, you go to one doctor for one thing, you go to another doctor for another thing. They don't talk to each other. There's no unified health record. So if you entered a genetic study and we wanted to know, did you ever have this problem? Did you ever have that? If you couldn't remember, or your parents couldn't remember, and we didn't know about that, we would mark that as a negative phenotype. So it's a really important, interesting question that has a lot of different dimensions, both from the standpoint of science and medicine, but also from the standpoint of what we really know about individuals that come into our genetic studies from the fragmentary records that we get from their current doctors or their parents or other family members.
I see. My last question actually is because I noticed that in the very last part of the number one disease that you mentioned, I mean, obviously there's the T cell phenotype in terms of Th17, dysregulation. Actually, I want your comments that do you think all these T cell dysregulation in terms of differentiation or T cell phenotype in the patient is because of the secondary effect from the epithelial TNF, or it's directly effect from this RAM1 and RAM2?
Oh, I think it's a combination. So in that case, there within the T cell itself, the ability to process different cytokines that require, and there's just dozens of them that require this kind of processing, are going to be impaired. But also, you point out a really interesting thing that is hard for us studying humans to address, but would be very natural for someone studying, for example, mice, to look and see how is the development affected in the thymus during, if you have this impairment in epithelial cells, or in this case, the IROM2s in dendritic cells and things like that. So I think, they're very hard to answer, those kinds of questions. And every now and then when I give a lecture about a disease, one of these diseases, someone will say, well, how is thymic development of T cells? Well, unfortunately, we can't go and take a kid and subject them to surgery and take out their thymus and answer that question as much as we would like to. Also, we often wonder what happens if you were to mate one of these kids with this genetic defect with one of our other genetic defects. But again, we don't have any easy way to do that. But I think it's a really important question, and my answer to that would be, I'm sure it's a combination of those effects in the end.
Okay, thank you, Mike. I think the other question from Hilaro Brazil. Do you want to ask a question yourself? Okay, I think I'm gonna just hello. Okay, I'm going to just read the question. So I'm not sure which particular disease he's pointing to. He asked if, I guess either mutation, the CD55 or the RAM12 can lead to other infections or any other bacterial. I mean, I guess he asked these patient or correlate or associate with the bacterial infection, either way with the RAM1, RAM2, or the CD55. Deficiency.
Yeah. Well, CD55, okay, so let's start with iris disease. So with iris disease, I have no doubt there's susceptibility to all kinds of infections. And I think we're going to see that as the patient population grows. So this paper will just be coming out. It was accepted recently at Nature Immunology, so that'll appear in the literature. And then what'll happen is other physicians, other research groups that do this kind of work will begin to focus on that gene, and we'll see more cases coming to light. And I'm quite confident we'll see different kinds of infections. And again, if we were to go back, which is difficult to do because of language issues and other things, for us to go back and actually query the families that we've already studied, we would find evidence of other infections. In terms of Chapel disease, they have low immunoglobulins. So they're like a hypogammaglobulinemia type of disease, for example, combined variable immunodeficiency, Bruton's agammaglobulinemia. So they're definitely going to get the whole range of diseases that you can get when you have low immunoglobulins. So I think that the answer to the question is clearly yes. They're going to be susceptible to other kinds of pathogens. And they're also going to respond poorly to vaccination. So I think, in terms of fighting off virus infections, they could have problems.
Okay. Okay, I guess I have one more question correlated with this second part of CD55 deficiency, that when you actually treat the patient with the drug, afterwards you actually examine most of the parameters with immune regulators. So I'm wondering if after the treatment, if you ever look also the T cell and B cell response with these patients, whether these are actually back to the normal level?
Yeah, it's interesting. We haven't done a lot of that only because the major parameters that, for example, the FDA wants to see in a clinical trial or that matter for the nutritional status of the patient are different kinds of parameters. So we haven't gone back and done a lot of extensive testing on the immune cells. But now that you mention it, it seems like we should really go back and try to study that to see if we could find that the overall metabolism of the individual has a positive effect on lymphocyte, for example, reactivity, due to just simple T cell assays. It's a really good, so in other words, would a T cell bathed in a starving person work worse than a T cell in a person who's fully nutritionally competent? And we could do those experiments. So I think it's a really good idea. I just can't think of a really good controlled experiment that we've done that would address your question directly, mainly because we focused on what we think is the thing really making them sick.
Sure, but I'm actually haven't read enough literature about the disease, but I'm sure if there's any mouse model with the CD55 deficiency, or the mouse assemble the similar phenotype as the patient.
Yes, there is a knockout mouse, but we don't see that phenotype.
Oh, okay.
So I don't know whether that's because it's the wrong strain, or because they don't get the gut pathogens or that cause that disease. But it really doesn't look like Chapel disease. So you'd say, well, maybe that CD55 is not the right gene, but now we have so many Chapel disease patients. I would say around the world, there's maybe 100 patients, and they all look pretty much the same. But again, not identical, right? Very similar, but not identical. Some have the diathesis for thrombosis, have severe thrombotic complications. Others don't have that. So I'm not sure why the CD55 mouse doesn't quite recapitulate the disease. But again, this is why we need human immunologists in this study.
Absolutely. Okay, so I guess if there's no other questions, we want to thank again Mike for the really fantastic talk. Thanks so much for doing this for the community. I mean, we all appreciate it. This is really great talk. So Lee, you want to say goodbye to Mike?
Yes.
Well, thank you so much for the whoever hosts this and puts this together. It's a really impressive forum I didn't know about before until Chuan told me. So thank you very much. I'm glad to participate, and Shesheni.
Thanks, Mike.
Thank you very much, Lee. Thank you, Mike. Have a good time in Florida. Enjoy the wedding. So I hope you all see you all guys next time. Thank you so much.
Okay. All right. Cheers.
Bye-bye.
Bye-bye, Mike.
Okay. See ya, Lee.